स्मेलिंग का शुगर एंड हनी चॉकलेट। मेरी बहन बोलती है उनके टाइम से परफ्यूम मैंने को बोला था दीदी वो शरद अंकल आते ना तो इस रूम में फुल फ्रेग्रेंस होता है। ये कैसे भैया आते हैं मेरे रूम में इतना अच्छा मेरी बहन बोलते शरद अंकल का परफ्यूम बना था वो बढ़िया फर्स्ट बेंच पे बैठती थी वहाँ पे इतना अच्छा स्मेल आता है जोर से आता है लेकिन पता है पूरा हॉलवे में आता है जब वो आते जाते हैं उनको एक बार पूछेंगे बढ़िया ओके भाई शरद अंकल का देख के लिसन पिछले क्लास में हमारा स्केल अभी भी होगा ठीक है ना अब भी जाएगी ना स्टेज फालतू में आओ छोटे भाई भाग भाग के आ गए हो प्रणाम हाय भाई हमें अलाउड है शॉर्ट्स में मैं भी पहन के आती हूँ एग्जैक्टली मेरी बहन ने जो सेम सेम पहन के मैं कैसे पहन के आ रही हूँ बहुत अलाउड है इसे बोला बाल खुले रखो ना उस पे चढ़ जाती है सही में हाँ इस पे चढ़ी है मुझ पे चढ़ी है अभी देखो मैं कितनी संस्कार सच्ची में भाई मेरे को आइडिया नहीं था मेरी बहन ने बस मना किया है मेरी बहन ने मना किया है तू शॉर्ट्स पहन के पैक कर दूंगी तेरा मैं क्या हुआ मेरे वाले में बोला शॉर्ट्स पहन के मत जा मैं पहनती नहीं हूँ फुल पैंट्स मेरा इशू है ये मैं लेगिंग्स में घूम रही हूँ ऑब्वियसली मैंने सोचा नहीं होता मैंने कितना ऐसे ना सोचा था क्योंकि मैंने इतने दो शॉर्ट्स रोज पहने बस दो पैंट शॉर्ट्स एग्जैक्टली मैं पहनती ही हूँ मेरे पास फुल पैंट्स के नाम पे इनके 3 पैंट्स है एक फ्लेयर वाले एक लूज वाले और 2 लेगिंग्स सेम है मैं वही पहने आई थिंक जिस दिन शुरू जब से 2 लेगिंग्स पहन रही हूँ मैं इधर आती हूँ हेलो हाय कौन बोल रहा है यार भाई तुम लोग मेरी तो खुद की बहन है मैं ये भी नहीं बोल सकती कि मुझे पता नहीं है मैं बोल सकती हूँ मेरी बहन क्यों नहीं अलाउ करेंगे शॉर्ट्स में समझ नहीं आ रहा मतलब रूल है या मतलब वो अकेले उनका ऑब्जेक्शन है कुछ तो होगा नहीं कुछ तो है इनका तो सब लोग गर्ल्स तो नहीं सकते हैं और काउंट नहीं करता एक्सक्यूज ऑन क्या कल इंग्लिश क्लास में मैम ने पास्ट टेंस का वो सिंथेसाइज करके डेफिनेशन देते हैं ना वो प्रेजेंट टेंस में लिखी ना उन्होंने डेफिनेशन की दिस इज अ फैक्ट तो वो प्रेजेंट टेंस में नहीं उसके जीवन में पता नहीं कौन सा हार्ट अटैक आया अगर आप पास्ट टेंस के बारे में बात करोगे तो बस पास्ट में अरे ज्योग्राफी क्लास में गांधी जी का फैमिली हिस्ट्री निकाल के बोल रहे हैं कुछ भी बोलता रहता है इसमें ना पहला इसमें भी बोल रहा है बहुत ओवर स्मार्ट बच्चा है ओवर स्मार्ट क्या बोलते हैं मैं बेटा ना हां कि वो इतना भी मतलब इसमें इसका मॉडल थे तो हाथ पे रह जाता है इसकी बेटी का ये भी हमने किया एंड सर ने ग्रुप को इसको भी बताओ थोड़ा एंड वो बताता है बहुत डिरोगेटरी आदमी है उसको ना लड़कियों से कोई इशू है मैं उसकी एक ही क्लास में बैठी हूँ एसएसटी वाला मैं उसकी एक क्लास में बैठी हूँ बाकी में मेरे पास तो कुछ ना कुछ काम आ जाते हैं वीकेंड से तो मैं आती नहीं हूँ ज्यादा उसकी ज्योग्राफी में आया तो हम तीनों थे इस कौन टीचर्स के अनुराग से बहुत जोड़ के हां एक टीचर ज्योग्राफी ऐसा मेकअप करने को आते हैं ना वो अरे वो सब ज्योग्राफी का लेते हैं एंड ही सेज रोज गण साक्षी ज्योग्राफी वाले सब के टेलिंग मी दैट स्टोरी हाँ told us this was like college में I had a girl who knows that teacher oh we didn't make a movement जय श्री राम बोलो जय श्री राम रुको भाई मैं बिहार को कॉल करके बताऊं कॉलेज में यार कॉलेज में एग्जाम में मुझे भी निकल गया था पर बोर्ड की तो मुझे नहीं गया मैं था वो भी नहीं चैट्स में एक्सपोज करेंगे आफ्टर दैट टेलिंग हमने किस योर वाइफ भाई आफ्टर भी कौन बताता है अपना प्रभात काल में अच्छे एडिटिंग एडवाइस मुझे क्लास को भी suddenly geography translation it just feels so good it's like different है तो क्या मूवमेंट था छोटे स्मार्ट ठीक नहीं होगा चलने
टेस्ट कौन आ रहा है?
मैं नहीं आ रही मैं तो कल ही जा रही हूँ। मुझे पेपर नहीं तो यार कोई संडे को मैं कल ही परसों मुझे पेपर चाहिए। बोल के आएगा सुनिए सी नहीं तो 8 है कि 0 है। 8 लोग क्यों देखेंगे? हाँ 0 है। इसीलिए ना मैं रुचा कोचा को मैसेज करती हूँ उसके ब्रांच से साइंस के टेस्ट के पेपर्स। सही है। कितना दे रहा है? कहाँ एड्रेस देने? भाई मेरे पेरेंट्स ही घर पे नहीं है, मेरा भाई अभी उसको लेकर आना पड़ेगा। लेके आ 7 साल का वो कूदेगा लिटरली। 7 साल के भाई की गर्लफ्रेंड नहीं, एक लड़की को पसंद उसके ऊपर कूद के। It's just how disappointing my life is. And then he's like तेरा तो हमारे ना disappointment तो मुझे विश्वजा को थर्ड के बंदे बहुत फर्स्ट डाउन करते विश्वजा आगे बैठना, सबको आगे जाना पड़ेगा तेरे चक्कर। अरे क्यों आना प्लीज मेरे को लेकर आओगे। आ गया थैंक यू। तू बोल रही थी साथ में वो तो मैं बोलूं दादिया की वो जीरो ने चला ली। No, like has an issue with your gender. She hoped I was a boy and now she gives me like intense tanas. No, she doesn't give me tanas, but अच्छा नहीं दिखता नहीं हमारे ऑब्वियस किया जाता। मेरी भी गलती मैं कहना हूँ। इतना ज़्यादा फेवरेटिज्म मेरी अगर कजिन की मेरी बड़ी बहन को चलता है क्योंकि वो फर्स्ट डॉटर है पूरे घर की। मेरा क्या फॉल्ट है? मैं दूसरे नंबर पे क्यों आ गई? या तो आती दादी को घर जा करती वो भी तो लड़की है। Dude, what do you expect from someone who never mind. Let me just kill her someday. That would help. Exchange में you have to get SND24. Yes.
How is that extra?
She'll tell you to sit in.
नहीं मेरे को फर्स्ट पे नहीं बैठना चाहिए, मैं आगे पीछे बैठेगा।
बहुत गंदी है। मेरे को नहीं पसंद फर्स्ट बेंच पे बैठना।
मैं पूरे साल फर्स्ट बेंच पे बैठी हूँ।
गाइस मैं भी पूरे साल मैं स्कूल में फर्स्ट बेंच पे बैठी हूँ। इस साल मेरे को बुद्धि आई है कि टीचर की चाकरी छीन लेता।
नहीं मतलब
मैंने पकड़ लिया सेकंड लास्ट बेंच।
मैं मैक्स टू मैक्स थर्ड लास्ट बेंच पे लास्ट सेकंड अरे मेरी
पर पीछे आया मुझे
यू पीपल वांट टू सिट इन द बैक। फिर खुशी है मुझे पीछे आगे भेजना।
हाँ एक्चुअली
खाना चाहिए यार।
Good evening. How are you all after a long holiday? You all enjoyed your holiday? Yes, ma'am. Huh? Yes. And forgot everything that we studied? No. So what we were studying? We were obviously science. We were studying. What we were studying, we had been talking about the chapter on cell. We had completed the plastics. So what are the organelles we had completed? What are the organelles we had completed?
cell membrane.
Cell membrane we are done.
Mitochondria. Endoplasmic reticulum.
Body, lysosomes,
nucleus, cell wall,
nucleus, cell wall, and plastics. So plants, animal cells we are done. I think only the plant cells. Diagram we had drawn. Yes, diagram we had drawn. So that means only the one part of the chapter, that is about the concentration. That part is left. So that we will learn today. So first of all, how do I understand that you all remember what we had studied? How do I understand? You have come after 3 weeks of holiday. Obviously, all must have evaporated already, right?
Where did you all go for a holiday?
Delhi.
Wow, good.
Himachal.
Oh, wow.
Mahabaleshwar.
Oh, nice places.
Going tomorrow.
Going tomorrow. You wanted to come for the class today? Just to show your face. What about you?
I came here and this is my first year in Mumbai, so I was going on to
Acha, you were not here before. From where did you come?
I came from Hyderabad.
Okay. What about you?
I went to village.
Wow, good. All of you had a refreshing holiday, no? So now let me give you a warm-up test first of all to understand how much you remember. All warming up gone. So let us have a short test. 10 questions I'll give you. See how much you remember. We are already finished with a major part of the chapter. We have already finished with the cell organelles of both plant cells, vacuole, plastics.
We have to still complete vacuoles and have the explanation of
Plastics and cell wall band. So that means little bit is left and the concentration. So that we will do and a lot of questions we'll have to solve. So just take a short test. Let me see how much you all
No, the
know.
video was revision before the class.
No. I am supposed to test you how much you all have, what did you all understand and how much you remember. Not to worry. This type of test, get attentive to write this type of test just to remember, recollect, or whatever you have studied earlier. No? Write down the first question and dictate the questions. You may write down those questions, so that will remain also as a set of questions for you. Number one. Name the organelle. That's the first set of questions I'll give you. After that, of course, we'll solve all the questions from the volume, your textbook. We'll go through one. The first set of questions you may write. Name the following 5 questions. Number one. So keep writing this way. Number one. Name the following. The organelle which produces enzymes. Organelle which produces enzymes. You may leave one line in between to write down the answers later. I'll give you some 5-10 minutes time. Number two. Part of the animal cell. Part of the animal cell made of part of the animal cell. I already gave you number one. Okay, I will give you the questions. You keep writing. Question and answer both. Keep writing. Question and answer both.
And this will give us time for the answer.
Of course. But not the entire period. 10 minutes. You have to become fast now, huh? Become fast. Answers here are hardly one line, one word. Or in case I may give you a question which would demand only a sentence. You don't have to write such lengthy answers, no? I'm just giving you some basic concepts, nothing else. Do you remember all these?
Few, a
Very
very
Few,
few.
huh? Those of you will be able to answer 50%, good enough. Chalo, try. I'm just giving you one word to write. No sentences even.
Mom, what's the fifth question?
Two types of endoplasmic reticulum. Two types of endoplasmic reticulum. In this first, excuse me, in this first 6 questions, you don't even have to explain anything. You don't go for explaining anything. Write only what the question is asking you. This answer should not be more than a sentence. One sentence is enough. Okay? Don't write unnecessary explanations and make your answers long. That's not the trend of the day. You should write only as per the question. Points. If it's a question which you know that there are 2 points to write, write in points, 2 distinct points. That way.
That's it.
Right. I'll give you exactly 10 minutes from now. All of you should be
Now we
finishing.
have to write all of the questions as well as the answers.
This would include at the most answers in 1 or 2 sentences, not more than that.
Do
One
you have to
sentence.
write the questions?
Huh? Questions up to you. If you want to write, you write. Those I asked you to write question and answer both, but if you feel that it's becoming too much for you, it's okay. We will discuss the answer. So when you keep the set of questions, good for you. So you know which question we discussed. Later you will not understand which answers you have written for which questions. In 10 minutes, before 10 minutes, all of you should finish. Keep practicing the diagrams of the plants and animal cell. As and when we move on to the next chapter, anytime I may ask you to this way, solve 1 or 2 questions, draw 1 or 2 diagrams. Anytime I may just give you some 5 questions to solve. So keep reading the previous chapter, whatever content you already finished. So that way, the more you revise, the better for you. And this is the time when you can, because by the time your school reopens, they will talk about a test now, the first exam, which will probably happen in July, no, in your school. So for that, maybe this first chapter would be there. This first chapter would be there. So get well acquainted with the chapter. Read the contents. There's a doubt. Finished? How many questions you don't know?
I did it 4, I think.
Oh, you did not get 4.
No, I did 5 or
You got 5?
yes, sir.
I got 7.
7. Then
Let me see.
I'm not getting 3 questions.
3 questions.
Not 3.
3. Decide. 2. First bench here.
Not 3.
3 not getting. Chalo, I'll give you another 10 minutes. I told you I'm giving you another 4 minutes. Another 5 minutes at the most. Chalo, fast. If you don't know, not a problem at all. You don't have to get stuck and remain that way. Whatever you know, you answer. We are going to discuss all the answers as it is. Whatever you know, you answer. It will be a self-check. I'm not giving you any marks. There's no competition. We are only trying to find out how much you remember after a long break. It has been a long break that we have had. So let's see how much you remember, but you have to keep studying. You have to keep reading.
No, I don't want to.
Very good.
I don't know.
Huh?
I don't know.
This to also try to think. Try to go through the question, see if you still remember something.
One is the answer. I remember Robert Hooke, but he did not propose the cell theory. He was the one who discovered dead cells. That's the only
Who
thing stuck
remembers
in
this
my
answer
head.
number 3?
I think I remember. I'm not sure, but I tried to.
Who are they? 2 scientists.
My president.
Clear
One Sweden
and
is strong. Sweden and strong.
What did you write?
I wrote Sweden and clear.
Check the spelling. Spelling should be correct.
Sweden and
How
strong.
do
Right?
you remember the names?
I'm pretty sure Sweden and strong.
You remember that?
That is the answer.
Sladen and Shwan. Sladen and Shwan. The entire complete name, if you remember, full name, if you remember, very good. Full name, if you don't remember also, but try to make an effort. I'm not giving you an alternative.
We just need to
You
remember.
have to remember. Full name likhna hai. Exam mein, in the exam, you have to write the full name. So what is the full name? Who is still writing? You want another 1 or 2 minutes to try? Or should we start discussing the answer?
No, completely.
All done? Ho gaya? All done? You wrote whatever you knew. This side, you came late, no? Did you answer? Some? How many more?
But I answered too.
You finished
I'm
only?
doing.
Okay. How many more you know from here? Tell me. Do you need time to write, or you want us to discuss? Start discussing. Acha, chalo. Now that we are done with this chapter, almost a major part of this chapter is over. Did you go through the questions of the volume? How many of you have gone through the questions of the volume? Did you try solving?
I did the first chapter. It's the other one.
Is it the first chapter?
Like in science, the other chapter.
Through the question answers, like the given
Science
question.
the chapter meaning?
Like
Other subjects.
Yes.
No, I'm talking about this chapter. Did you go through the?
I went through answer the following that they had provided.
So see here, there are so many questions that are there in this chapter, in the module. You have to go through the questions of the NCERT book. That is obvious. There's no option. You have to go through that. But not necessarily you will always get questions straight away like the book in the paper. Some few questions will be exactly what is there in the book, but most of the questions will not be there. Will not be exactly what is there in the book. So you have to learn the correct way. Now that you are heading towards the 10th standard, you will not get questions which are exactly there in the book. The concept you have to know. And questions you should be able to answer on your own. So develop that method right from now itself. The sooner it will be easier for you in the 10th standard. Otherwise in the 10th standard you will have a difficulty in framing answers. So now that you are feeling quite happy that I'm in the 9th and now next year like you in the 10th, answers also has to be smart enough, no? The way you study also should be smarter enough. The way you write answers also have to be smarter enough. So what is the right method of study now? Read the chapter in and out. You have to read the chapter very nicely. The volume we will refer for writing answers because in the volume we have given everything in points. Functions, structure, what are the points for the structure you are supposed to remember. So that's how I teach also. Every point you have to remember. The differences, what to write, etc. etc. Okay. So let us discuss some of these questions. What is the answer for number 1? No, number 1 is a different. I did not write number.
Organelle which produces enzymes.
Enzymes.
This one I didn't get.
Yeah.
But what is the answer? Who knows this answer? Who knows this answer? I need to know all your names.
This is the one, the two I
Pata hai?
This is one of the 2 questions I did not try.
Organelle which produces enzymes. Which organelle produces enzymes?
So this is one of the 3 questions that
The all same answer all of you will say. Not necessarily, no. Always go out of the box. Organelle which produces enzymes. How many organelles you have studied? Mitochondria. Does it produce enzymes? Nucleus. Does it produce enzymes?
No.
Endoplasmic reticulum
Golgi
also no.
bodies.
Golgi.
I knew it. Why did I not learn it then?
God has given us such a big structure. Use every part of it.
And the answer is gone.
Yeah.
So function-based study, Golgi produces so many different types of enzymes. What is the next question now? What is the answer for this?
I think it's endoplasmic reticulum.
Endoplasmic reticulum.
Not necessarily what others are saying, you also have to say. Always go out of the box. What is this?
That's fine.
Cell wall.
Huh?
Cell wall.
Cell wall. Animal cell में cell wall. कभी मैंने पढ़ा है animal cell में cell wall.
Cell membrane.
Cell membrane.
Cell membrane. Yes. That's what I meant.
What is the other name for cell membrane?
Plasma membrane.
Plasma membrane. ठीक है? Got the answer? Those of you got it wrong, correct करो. Do the corrections.
Wall instead of membrane. So it's
Cell membrane or plasma membrane. Number 3. This is very easy.
I didn't remember the word cell membrane.
Number 3.
Mitochondria.
Mitochondria. Number 4.
Golgi bodies.
DNA.
Chromosomes and DNA.
No chromosomes.
I
DNA.
wrote DNA/chromosomes.
No, this slash ka method abhi nahi chalega. You'll get both the answers wrong. No options. Don't give the teacher to choose options from both the answers and then teacher will give you mark. We will just straight away cut it down. Finish. We are not going to choose answers for you. No? Our duty is to teach you. Even in your school, they will not choose answers for you. No, come out of this habit. Don't write this because most of the students, when they are confused, वो option दे देते teacher को, तुम ही choose करते रहो. We will not choose. Just कर दो. Don't write two answers for any question ever. So that's how I'm repeatedly telling you, revise karna hai, read the chapter very nicely. Not to take stress, little section of the chapter you should read. Today I will read mitochondria and plasma membrane.
What was the answer?
Which one?
DNA.
DNA, okay. 5.
Rough endoplasmic.
And smooth endoplasmic reticulum. 2 types I asked you. Number 6.
Bacteria cells.
Type of cell to write. Example I did not ask you.
Prokaryotic.
Prokaryotic. Type I asked you. So type would be do not possess a well-developed nucleus. Prokaryotic.
Yes.
Corrections? Keep doing. Why is the cell membrane known to be semi-permeable?
Does not
Because
allow
it
all
allows
substances to pass through.
It only allows selected substance to pass through.
On this question, I didn't say.
What should the answer be? Why is
Selective substances to pass through.
it allows certain substances, selective substances to pass through and does not allow some substances.
I wrote, does it allow all substances to pass through it? Therefore, it's a semi-permeable.
So it does not allow certain substances to pass through, or it allows certain substances to pass
Therefore,
through.
it's
Both the
partially
answers are
permeable.
correct.
I didn't remember what semi-permeable means.
Semi means, understand from the word, semi means half, partial, no? Why are leaves green in color?
Because of the
chlorophyll
present inside the plastids.
Because of the presence of
Chlorophyll in the plastids.
because of the presence of chloroplast which produces chlorophyll. You have to name the organelle because the chapter is all about cell organelles. Your focus should be on the cell organelles. Whenever we are correcting answers on this chapter, the objective of teaching you this chapter is to make you aware about the cell organelles. So that part is always looked for in the answer. You should know the cell organelles, write the cell organelles. Why the leaves are green? Because of the presence of chloroplast which produces chlorophyll. And now this Schladen and Schwann. And what is the full name is what? What is the full name? Who remembers? Full name is? Schladen, Matthias Schladen and Theodor Schwann. Okay, so this you'll have to remember. Chalo, now let us continue with the chapter. Keep reading. Off and on, I'll give you this
I actually
short.
got both spellings.
Huh? I
actually got both spellings correct for the last name.
both spellings correct.
I thought I got them wrong.
Achha, very good. If you're, so when you learn the right way. So there's no option that you'll get it wrong ever. When you are learn the right way. So what are we talking about? This particular chapter. What are we left with is we are talking about certain features of cell membrane again. Cell membrane, the basic structure function we had studied already.
Yes.
Cell membrane, the basic structure function we had already studied. We had already come across that if it is the cell membrane is made up of lipids and proteins, etc. And then the functions would be the passage of certain substances. I gave you one question also that the cell membrane would basically help in the movement of certain substances and hold certain substances from transport across. So here we are understanding some other feature of the cell membrane, that is what is known as permeability. That is what is known as what? Permeability. Now what is the meaning of permeability? The ability of any cell, any membrane to allow transport happen. Allow transport happen. So we are majorly talking about the transport functions of the cell membrane. To understand this, we have to understand a bit more. We are not only talking about writing down the function. Permeability we have to understand in certain specific conditions. Now listen to me very carefully, try understanding. What I'm doing is I am taking 3 respective beakers, okay? I am taking 3 respective beakers here, and in each of these beakers there is a specific solution taken here. There's a specific solution. The solution, when I say, you are clear that there is water and there is some other solute. So the moment I am talking about this word solution, the moment I am using this word solution, you should very clearly remember, I am talking about water and certain solute. I am talking about water and certain solutes mixed into water. Now what can be the solute? What is the example of solute? Solute can be salt, solute can be sugar, solute can be any type of substances that easily get dissolved. So basically, to give common examples, we think of sugar, salt easily getting dissolved in the water. So that is the basic concept of solution when I say. What next? We are talking about certain cells that we take in this solution. We talk about certain cells. Now, since we are talking about the cells, let us use the concept of plant cells only. Recently, we were talking about plant cells. So, I'm talking about plant cells. Keep some plant cells in this solution. So, what are we doing? We take some plant cells in this solution. We are taking some plant cells in this solution. Now, right now, don't be bothered about the structure. We are not talking about knowing the structure right now, but only one particular parameter, that is the vacuole and the sap. That is what we have to understand here majorly. What is the meaning of sap? Did we study that? Vacuole we did not study? No. Achha. So if vacuole we haven't studied, then we'll take up the vacuole first of all. For a plant cell, what are some of the structures? Check the diagram that you have drawn about the plant cell. The other day we have finished.
I believe we only have to cover vacuole. Just everything we had covered.
Plastids we have finished.
Yeah.
Plastids we have finished. Cell wall already we have finished. So only the vacuole to talk about. Check the diagram that we have drawn. Plant cell. If this is a plant cell, then obviously it should be having the cell wall and the cell membrane both. And there is a vacuole that the plant has, that the plant cell has. Now, what does the vacuole contain? It contains water and dissolved salts. Various different type of substances dissolved here. So, this is the sap that I am talking about. This is the sap that I am talking about. Understand what is the definition of the word sap? Sap always means water and salts dissolved. So, a variety of salts, enzymes, etc., hormones, etc., all remains dissolved in this water. It is not just clear water that we can talk about in a plant cell, just something like the blood that we have in our body. In our body, we have got the blood, which has got a whole lot of substances dissolved. So, same like that, that acts as a fluid in case of the plant cells. Now, we are talking about the sap. Now with that relation only I am talking about, but let us just learn 1 or 2 features about this sap. So I already told you what does the sap contain. The sap will always contain water and variety of salts. But what do you think should be the function of this sap or this vacuole? The function of the vacuole, if I am talking about that the plant cells always have got vacuole. Then what should be the function of the vacuole? Number 1, to maintain the shape of the cell. Yes or no? Of course. Now that I talk about the vacuole is always known to be responsible for the shape of the cell. Now how shape of the cell we can understand, because I talk about a leaf which is so fresh. Are we not able to understand a difference between a dry and a fresh leaf? Of course, no? We are able to understand a difference. And In case of a fresh leaf, you will understand the vacuole is filled with water. A plant which has got fresh leaves, we understand the sap is adequate in the vacuole, meaning the vacuole is not dried out. The vacuole is abundantly filled with water. So yes, shape, the vacuole and the sap provides shape to the plant cells. We also say that it provides rigidity to the soft plant parts. Do you understand the meaning of rigidity? What is rigidity? Rigidity means some stiffness. Stiffness. Huh?
Solidity.
Sturdy.
Aha. So when I talk, not solidity, solidity is not a word at all to be used here. So now that we are talking about that stiffness, I would say, a more appropriate word. Stiffness, I would understand that the green stem of the herbs. Say suppose I talk about coriander, I talk about small herbs like this. When the coriander stem becomes dried out, is it stiff? No, it is limping. It is limping. I talk about any particular flower sticks. When the flower is running short of water, what would happen? The stem would limp.
Yes.
We understand that the cells are running short of water. But when it is fresh enough, stiffness of the plant part. Now I'm talking about, yes, this is the sap which is important in maintaining the stiffness of the plant part. Any particular potted plant that we have, by morning when you observe them, you will see that all of these leaves are fresh and then the stem and everything stiff enough. But by afternoon nowadays when it is so hot, you will see that quite a number of them are becoming softer. They have lost so much of water, that's how. So yes, when I talk about the sap, what is the sap responsible for shape? What else? The sap is also responsible for the storage of the food. How storage of the food? Because now this sap, now that we are talking about this sap, the sap is also dissolving the glucose that the plants have produced by the method of photosynthesis. So yes, storage of the food also. Now with reference to that, with reference to this plant cell, we try understanding certain words. One is known as turgidity. You will come across these words written there in your book, in the volume. Turgidity and flaccidity. Now when I talk about turgid, try understanding what is a turgid condition. What is a turgid? A cell which has got large amount of water. The cell is tight filled with water. Turgid. And what about the flaccid condition? Flaccid means a cell lack of water. Just the opposite. A cell which has lack of water. Do we understand in a plant? Yes, when the stem is limping and becoming wrinkled, we understand dried out. So we understand, yes, the cells are flaccid now. Okay, so with this concept, we are coming across. In this solution, what are we adding? What are we putting? Some plant cells. What are some of the characteristics of the plant cells? Let us understand. What would be some of the characteristics of the plant cells? So now these plant cells that I understand here, this plant cell, the first plant cell that I have put here, would be having a vacuole. We'll not talk about the cell wall, cell membrane, all these aspects. We are only talking about the vacuole. This is what we try to understand in this topic. Okay, and then what about the second solution? Yes, here also we have taken some plant cells. It should be having the vacuole, it's a plant cell. In the third also we have taken some plant cells. It should be having the vacuole, it's a plant cell. So what are these? We are talking about three beakers, something like this, A, B and C. We are, listen carefully. Concentrate in the class. Don't start dreaming. They will not understand anything what I'm speaking. So now we are talking about in the plant cells. We are talking about plant cells. We keep in a solution. I again repeat, we are not understanding the strength of the solution. Now, what is the meaning of strength of a solution? I may talk about 20% solution. What is the meaning of this?
There's a 20% of the solution.
20% solution means 100, if it is 100 ml, say suppose a solution. in this solution, in a scale of 100, I would say, in a solution, 20% means 20 parts solute and 80 parts water. So total 100 ho gaya na. So I'm talking about 20. Similarly, if I talk about 40% solution, then what do you understand? 40% solute, 60 parts water. Okay? This concept you should be understanding. So now I am saying that we have been given 3 beakers, solutions already there. We don't know what is the strength of the solution. We are only asked to put some plant cells and go for an experiment.
Can you please turn on the fan?
So now we put these plant cells there and try for an experiment. So what are we doing now? Wait for certain periods. Now say suppose wait for some 6 to 8 hours. After 6 to 8 hours, what? Observe whether these cells are showing any change or not. So in the first beaker, we get to see that the cells do not show any change. The cells are just as it is. After this period of 6 to 8 hours, the cells in the first beaker do not show any change. What about the second one? No, in the second one there is a change. What are we observing? The cells which are in the second one, the cells have become something more bloated up like this, bigger than before. The cells have become bigger than before a little bit. And what about the vacuole? And if I'm talking about the vacuole has become big enough. The vacuole was not this big earlier. It has become big enough. So I would say, what happened to the cell? The cell became swollen.
It got more water.
We'll learn what are the factors, what has happened, and the conclusion. What about this? In this case, the cell has shrunken than before. The cell has shrunken. 3 different conditions. So I'm talking about in the first, what has happened? The cell remained as it is, did not show any change. In the second, the cell swelled up. In the third, just the opposite, the cell became shrunken. What must have happened here is, when I talk about this particular plant cell, in this plant cell, why did the vacuole remain as it is? What must have happened? The vacuole will remain as it is, the cell will remain as it is only when whatever amount of water molecules are entering and whatever amount of water molecules are leaving the cell are almost the same. Almost the same. For an example, I may understand that obviously the molecules of water will be entering and leaving. We cannot say that nothing is entering or nothing is leaving. A whole lot of water must be entering the cell. Yes, a whole lot of water must be entering, a whole lot of water must be leaving also. But if the net movement is 0, meaning if 10 molecules entering, another 6 going out. If 12 molecules entering, another 13, 14 going out. So is the cell able to maximally gain anything? Is the cell maximally able to lose anything? No, the loss and the gain is almost the same. So we would say the net movement of the water molecule is 0. The cell has neither gained anything much, nor the cell has lost anything. And that is one big reason why the cell shape did not change. That is one big reason why the cell shape did not change. The cell remained as it is. Now our concept to study is not only about the cell shape, but we are also focusing on what type of a solution is it, and we have to compare with the cell sap inside.
And can we say the solution had adequate water?
Listen first, try understanding first. So now, when we are talking about, first you learn the terms, then you talk about your queries. First you learn. So now that we are talking about then what type of a solution it is, such type of a solution. Remember, we are defining the solution with reference to the sap. That's why we have been talking about the sap that side. So this type of a solution, then what is the name of such type of a solution? Such type of a solution is known as isotonic. From the word iso, refer. Iso means same. What are we comparing? We are talking about that the solution here and the sap here has the same concentration. But you may ask that how is it possible? Because outside in the beaker there must be so much of water. How can we compare that much of water with a small cell? And how much of the sap must a cell be having? How is it comparable? We are not comparing that way. We are not comparing the small amount of sap with the water outside. We would compare 1 ml of the sap and 1 ml of the solution. Something like, can I compare a 9th standard student with a 5th standard? Of course not. If I have to conduct a competition, I have to take a 5th and a 5th or 5th and a 6th. And the 9th and the 8th or 10th and the 11th, like that. So that is the exact thing. You cannot understand a comparison between a whole lot of this solution and the small amount of sap. Of course not. We are talking about 1 ml of this and 1 ml of this. And then see. So when we took 1 ml of this and 1 ml of this, it was found that yes, the concentration was almost the same. What is the meaning of concentration?
The amount.
What is the amount of water and what is the amount of solute? And it is found that again I use the word concentration, get acquainted with these words. So I am talking about that the concentration of this solution here and the concentration of the sap is exactly the same. It was almost the same, it was found. And that's the reason the cell did not show much of a change. Then what happened in the second one? What happened? Why did the cell swell up? Then what do you think? What should have happened?
The
concentration
of
of
water in the
liquid
solution was larger
than
The concentration of the water, the part of the water
in the solution is greater
than the cell.
Compared to the cell.
In the cell, correct. And that was the reason, if I now try
So more
understanding
water was going in than was
so
coming out.
much of water. So now I talk about the vacuum. Why did the cell swell up there? Why did the vacuole become so big than previous?
More
The
water
vacuole
was-
became so big than previous because so much of water has entered. But can I say that no water must have left? No, water molecule will leave, but that was negligible. The water that has entered is much bigger than what the cell left or what the cell lost. The amount of water entered is much more than the amount of water that left the cell. And what is this process of intake of the water known as? The process of intake of the water is known as endoosmosis. Take a note of all these terms, you'll have to remember. What is this called? Endoosmosis. What is endoosmosis? The process by which the cells take in water. Don't write, I will give you proper answers, not to write. Understand, just samjho pehle. So now that we talk about that the cells have started taking in so much of water, that's the reason the cell now gradually started becoming swollen. Can you give me an example that happens in our homes or in our day-to-day life also? We have seen something like this.
When we soak in resins.
When we soak in raisins, when we soak in any type of seeds, when we talk about the grams and etc. that we soak in water, what happens? Did you ever observe? You put them in the water, the next day they become swollen. They have gathered so much of water. Did you ever observe this different type of lentils, the dals and all that we soak in the water, the different type of grams that we soak in the water? Before soaking, they are not dried out. You put them in the water, let them be in the water for almost about 12 hours. Now what happens? They have become bigger. So that means inside, every cell must have absorbed so much of water. And every cell has now become turgid. What is the meaning of turgid? Filled with water, full of water there. Then, yes,
So it's a process of swelling called endoosmosis.
Not the process of swelling, the process of intake of the water. Process of swelling is different. That is turgidity.
Endoosmosis is
Endoosmosis is the process by which water enters into a cell. That is the correct definition to write. What is endoosmosis? The process by which cell takes in water. The process by which water enters into a cell. That is endoosmosis. Clear? Everybody is clear about this concept? So this is endoosmosis. Now we have to also talk about then what are these type of solutions known as? The topic is basically about this. What type of solution? This solution, we will name it as hypotonic. Hypotonic. What is the meaning of hypotonic? Concentration lesser than any other comparable medium. So I'm saying that this particular solution is hypotonic. Also, the other word for hypotonic is dilute. So what are you understanding? I said this solution is dilute than the cell. Something like, again try understanding. When I'm talking about the word hypotonic or dilute, can I say that the cell is, can I say that this solution is dilute or this solution is hypotonic? Is that the complete sentence? The solution is hypotonic. The solution is dilute. Is the sentence complete? Actually not. I should say the solution is dilute as compared to the cell. We are comparing. The solution is hypotonic as compared to, we have to compare with something. The solution is hypotonic as compared to the cell. Something like, if you are not following, it's something like, if I give you an example, I say that Rohan is taller. Full stop. Is the sentence complete?
Then
I have to compare with somebody, you know, when I'm saying the word, when I'm using the word taller. So here exactly, the moment we are using this word tonicity, meaning we have to always compare with something. Immediately understand that yes, we are comparing with something and now we are comparing it with the cell. So I should say the solution is hypotonic with reference to the cell as compared to the cell.
And same with isotonic.
Now isotonic means yes, same with isotonic. Here also we are talking about the concentration is, the solution is isotonic as the cell. Hypotonic as compared to the cell. Hypertonic as compared to the cell. So now let us go forward. Is this understood? So if I give you questions, you will get questions in your paper. What happens to the cell when it is placed in a hypotonic solution? What should you write?
It will
The cell will
go through and
swell up.
And it will swell.
The cell will swell up because of the process of endosmosis. Now again try understanding one more thing. I am comparing the cell and the solution. Tell me, where is water more? Here or here?
solution.
In the solution. It is a dilute solution. What is the meaning of a dilute solution?
And more water.
More water, less of solute. Is this clear? If this is clear, then this will be very easy. Now I am talking about the cell is shrunken. What must have happened? That the cell is shrunken. The cell is shrunken because the cell must have given away so much of water as compared to what it got. The cell must have given away so much of water. Maybe now 10 molecules of water gone and only 2 molecules it got. So the gain is much lesser than the loss. So if this is what is happening, what is this process? This process is known as exoosmosis. So what is the definition of exoosmosis? The process by which the cell gives away water. The cell loses water. So the process by which the cell gives away large amount of water is exoosmosis. The process by which the cell absorbs or gains large amount of water is endosmosis. When will a cell behave like this? When will a cell behave like this? When the solution is much lesser than the cell. We are comparing, no? So now the cell must have given away so much of water only because the water is less here and water is more here. Again, you may feel that how is it possible? There is so much of water and you are talking about a small cell saying that the cell has more water. No, I have to understand 1 ml of the cell sap and 1 ml of the solution and then find out. Obviously, something like when I talk about the ship on the ocean, on the water, are we to understand that the ship, why is the ship not sinking?
Buoyancy.
Because the ship is now on the surface of the water because it is heavier than whatever amount of water it is dispersing. It is not heavier than the entire water of the ocean. Compare the entire amount of the water in the ocean and the ship, obviously the ship should start sinking down. No? So now when we are talking about why it is floating, because it is heavier than only that amount of water what it disperses, what it has in its surrounding. Exactly that concept. I'm talking about 1 ml of this, 1 ml of this. And every time we see if outside solution has got more water, then the cell will give away water or lose water. The cell will give away water or take water? Take. And now what's happening? The outside has less water. Cell has more water. So the cell is giving away water. What process? Exosmosis. What will be the name for this solution then? The name for this solution would be hypertonic. The name for this solution would be hypertonic. So what are the three terms we understood? A solution can be hypertonic, a solution can be hypotonic, and a solution can be isotonic. But we are comparing with the cell always. I had been talking about the plant cell. Isn't this applicable for animal cells? Very much. This is very much applicable. Now the same activity is also done with animal cells like the RBC cells. The same activity is done with RBC cells. Understood? The same activity is done with RBC cells. Now we have to remember the definitions of endosmosis, exosmosis. This is what is commonly asked in your paper. And then we have to talk about hypo and hypertonic solutions. Let us define each of these terms. Number one, we will write isotonic solution. We will define these terms. Write down the definition and various other type of explanatory questions also you will get. It is not only definitions that you will be asked to write. But let us first of all start this way. Isotonic solution. Isotonic solution. What should you write as a definition for isotonic solution? A solution which has equal concentration of solute. Equal concentration of solute and solvent. Equal concentration of solute and solvent. As a cell. Equal concentration of solute and solvent as a cell. We are only talking about a definition of isotonic. Come to the next one. Hypotonic solution. Hypotonic solution. Hypotonic solution. What will be the definition of hypotonic? A solution which has higher concentration of water. A solution which has higher concentration of water and lesser amount of solute. Higher concentration of water and lesser amount of solute. Lesser amount of solute as compared to a cell. Lesser amount of solute as compared to a cell. As compared to a cell. Understood this? Understood this? Then how should we write hypertonic? Tell me now. If this is the way we have framed the definition for hypotonic, now tell me what should we write for hypertonic? Yes.
A solution which has lower concentration of water and higher concentration of the
solute. Very good.
Solute,
so this is what
in comparison to the
in a cell. In comparison to a cell. Okay. Write down. What is a hypertonic solution? A solution which has higher concentration of
Low concentration.
I said a solution which has higher concentration of solute. Either of the one you can write, and a lower concentration of water as compared to a cell. As compared to a cell. Understood these three? Now, endosmosis and exosmosis. We'll write down the difference. Take one question. First, you complete the definitions. All of you, are you done with the definitions? Isotonic, hypotonic, hypertonic. Is this clear? Write down now the next question. Write two differences between exosmosis and endos. Any doubt?
Lower concentration of water.
What is hypertonic solution? See what you have written. A solution which has higher concentration of
Water.
then hypertonic. It should have higher concentration of solute and
Lower.
water as compared to a cell. Complete. Next we will do write two differences between endosmosis and exosmosis. Endosmosis and exosmosis. We'll complete this one. Then we will learn the difference between osmosis and diffusion. Make a table. Write down difference between endosmosis and exosmosis. I will only give you the points and you try framing your answer to write. You don't want the AC?
It's fine either.
Don't want? You are putting it off. Don't want? Huh?
It's fine.
Who came from Shimla? You said? Ah. She is wearing t-shirt, and why you are wearing all your pullovers and jackets? You feel cold? Okay. Differences between endosmosis and exosmosis. What should be the first point? The first point you will write on what is the process, meaning the definition basically. What is the process? So endosmosis, what should you write? The process by which water molecules,
Just a second.
process by which, did you make the table? Right. So first one, under endosmosis, process by which water molecules, process by which water molecules, process by which water molecules are, or process by which water molecules enter the cell. Process by which water molecules enter the cell. Due to the concentration difference, and permeability of the cell membrane. Now this is a format of writing. Huh? Read what you have written. देखो समझ में आ रहा है कि नहीं? Read. Process by which water molecules are entering the cell. Why should it enter? Because the cell membrane is semi-permeable and it is allowing water to enter. And concentration difference also will play a role. देखो यहाँ पे the concentration was same, so the water molecule was not much entering. Here the concentration is not the same. That is what the water molecule was entering. So the water molecule is moving from a region of high concentration to a region of low concentration always. The water is moving from a region of high concentration to a region of low concentration, and this is standard. The water will always move, or most of the substances, they will always move from a region of high concentration to a region of low concentration. Did you understand what you have written the first point? Endosmosis. Then what should you write for exosmosis first, tell me? What should you write for exosmosis?
Process by which water molecules are exiting the cell due to the concentration difference.
Process by which water molecules are
Exiting.
Exit is not a very proper word to write. Leave the cell or are given away by the cell. Process by which water molecules are given away from the cell or leave the cell. Process by which water molecules leave the cell and the rest part will remain exactly the same. Process by which water molecules leave the cell because of the concentration difference and permeability of the cell membrane. Another difference we'll have to write. Now I'll tell you the point on what, based on what you should write on the difference. Second difference. Second difference. Now, second difference, when will endosmosis happen? Tell me. When will
When the
endosmosis happen?
solution is hypotonic in
When the
comparison to the
solution is
when the solution is hypotonic in
hypotonic.
in
Understand?
comparison.
When will endosmosis happen? When the cell is placed in a hypotonic. Then when will exosmosis happen?
Hypertonic.
When the cell is placed in a hypertonic solution. Dekho, write on your own. When will this process happen? That's the second point you should write. Endosmosis. It will take place when the cell is kept, or endosmosis, this will occur when the cell is placed in a hypotonic solution. Whose phone is not switched off?
It's not ringing. It's not fine.
You all are supposed to keep your phone switched off. I think all of you are told, no, already. The moment you enter the classroom, no vibration, nothing. Nothing. You are supposed to just switch off your phones. In the break also, actually, you are not supposed to keep fiddling with your phones for that 5-10 minutes break. Okay, got that? Second point, did you all write? When will this process of endosmosis happen? Remember, hypotonic solution should be outside. When will this happen? Hypertonic solution should be outside. Then only exoosmosis. So what are we learning? We are learning one particular process right now. We have been talking about one particular process right now. What is this process now that we are talking about? Osmosis. We are learning osmosis. What are some of the examples of osmosis to understand? One of the most common example of osmosis is the process by which the roots absorb water. Osmosis. So if I'm talking about any particular plant, now that we have already been talking about the plant cells, I talk about a plant, and this plant has got so many roots, and the roots are constantly absorbing water. Talk about one single root cell. Why did it absorb water? Talk about one single root cell. Why did it absorb water? Because the cell sap is
It has low concentration.
more, the cell sap has more concentration, or the cell sap is hypo or hyper. Which one we should write?
Water travels inside the roots of the water because
My question is that why is the water entering inside the root cell? This is a hypertonic solution or hypo? Hypo. The soil water is hypotonic when you compare it with the root cell. Now I'm talking about that under normal condition the water is entering the root cell because the root cell is concentrated, the outside medium is dilute. That's why. But I put a whole lot of fertilizers here in the soil. Now I am putting a whole lot of fertilizers. Is the soil water still remaining hypotonic? Now it is becoming hypertonic. So what are the chances? The plants may die, no? if I put in more and more of fertilizers, etc., there are chances that if I talk about some small plants, herbs, etc., yes, within a couple of days they will die. But why are they dying out? Because now that I put in so much of fertilizer, the outside environment has become hypertonic, they have become concentrated. And now these cells have started giving water. The plant cells have started giving water. Will the plant cell give away water in this situation? Yes. When the plant cells have a dilute medium outside, they grab water. But the moment the outside environment starts becoming concentrated, they would give away water. And if the plant cells have to give away water, obviously they will die in a short period of time. So that is what happens whenever we spray too much of those insecticides and spray too much of fertilizers, etc. That's the reason instead of growth, the plants die out, start dying out. Okay. Now let us talk about osmosis. After knowing all these basic concepts, then what is osmosis? Then what did we study? What is osmosis?
Osmosis is the transfer of water from
Water molecule
to
lower. In all these three experiments, what we were studying? Water molecule only, movement of the water only from a region of high to low. From a region of high to low. So we were basically talking about the movement of the water. But instead of osmosis, if I have to talk about diffusion, I would also say that can I include osmosis in diffusion? Or is the concept of diffusion also including osmosis? Yes, because diffusion can be of various types, and one such type is osmosis. When I talk about diffusion, it's a bigger concept. Osmosis is comparatively a smaller concept. So what is the difference? I may talk about diffusion where I may think of gases, where I may think of solids, where I may think of liquids. But when I talk about osmosis, no, I have to only think of liquids and only water preference. Then what is the definition of diffusion? Let us learn. What is the definition of diffusion? The movement of molecules, try understanding, the movement of molecules of substances, the movement of molecules of substances from a region of high concentration to a region of low concentration. I talk about a beaker and then I take some water here, I drop some crystals of sugar, The moment I drop some crystals of sugar, yes, all of them, they settle down at the bottom. I put some crystals of sugar, some sugar cubes I take, drop them in the water. After dropping them in the water, I take 1 spoon of water from here and I taste. Is it sweet? No, it will not be sweet. Try it out at home today itself. This water will not be sweet at that moment when you have just dropped the sugar cube and take 1 spoon of water from here and taste. It will not be sweet. Wait for 10 minutes and take 1 more spoon of water. Now, yes, it is sweet. And even more, you stir it and then take 1 more spoon of water. It is sweet, quite sweet now. Why? What has happened in all these 3 stages? Initially, when you drop the sugar cubes, they did not start distributing. So the sweetness did not travel till here. In 10 minutes, what has happened? They started getting dissolved. They started moving. The molecules started moving. Yes or no? They started moving. And yes, after some time, when I take some amount of water, yes, I can feel the sweetness little bit. When I stir, what's happening? The entire thing is getting converted into solution. When I am stirring, the entire thing is getting converted into solution. What am I talking about? Am I talking about the movement of the water molecules?
No,
Or am I talking about the movement of the solute molecules from a region of concentration to a dilute medium? Am I clear? So I'm talking about here the sugar molecules were concentrated. So the sugar molecules are actually moving from a region of high concentration to a region of low concentration. So now I'm talking about the movement of Now I'm talking about the movement of solid, solute or solid. Can you give me an example of similar type of a situation with a liquid? Can we think some type of an experiment, some type of
Liquid
a
to two of them
mix.
liquid?
where they like mix.
Where they mix or talk about water again, a simple activity. I talk about water again, same way. Take water in a tumbler, put 2-3 drops of some ink, liquid to liquid I'm talking about now. Water, 2-3 drops of ink. What is happening? Initially, is that ink immediately getting mixed with the water? No. The droplets of the ink, you will see, first of all, start dispersing from the bottom and then slowly come up. Or from the upper layer, slowly go down. So now what's happening? These drops of ink, the moment you put, the upper layers are first colored. Then the ink is dropping, and slowly the layers of the water are getting the color. This is with liquid and liquid. For gases, can we ever think of migration of the molecules of gases like this? Perfume, very good, perfume.
The smell of food.
Smell of food. When your mother is cooking at home in the kitchen,
You can smell it from the other room.
next rooms also.
Incense sticks.
Incense stick. So what is happening? When I'm burning the incense stick, the concentration of the molecules of the gases are there, but they are moving between the molecules of the air and spreading the smell everywhere. All these examples that I have been talking about, I gave you examples of the solids, we have been talking about examples of the gases, we have been talking about examples of the liquids. But if I talk about osmosis, I have to restrict myself only to this. I cannot talk about solids and gases for osmosis. So now what is the basic difference? If I talk about diffusion and osmosis, what is the basic difference?
Osmosis takes place only in liquid.
If I talk about diffusion, we are talking about the spontaneous movement of molecules of gases, liquids and solids from region of high concentration to a region of low concentration always. Osmosis, movement of water molecules only and only water molecules. So that's why I told you that when I talk about diffusion, it's a big concept. I can talk about so many examples. Look, think of so many examples of gases we can think of, solids we can think of, liquids we can think of. But osmosis, no, only and only water, only and only solvent. Only solvent molecules. Let us write down the definition of osmosis and diffusion with one examples in each case. One examples in each case. Differences also you are asked in the exam. Let us write down the definition. Definition of osmosis. Or first you write diffusion. First you write diffusion. The spontaneous movement. The spontaneous movement of molecules of a substance. Spontaneous movement of molecules of a substance. Spontaneous movement of molecules of a substance, that is solid, liquid and gases. From a region of its high concentration, to a region of its low concentration, when the molecules are in direct contact. When the molecules are in direct contact. When the molecules are in direct contact. Now what does that mean? Molecules are in direct contact. What does that mean? I was talking about the concept of diffusion. I may say that if these are the molecules of the water, these are the molecules of water, and if I have to talk about diffusion, then what? If these are the molecules of water, what has happened? The molecules of the sugar which were concentrated at one end, they started moving in between. They started moving in between. This is what we will understand always as diffusion. What did you write the last part of the sentence? When the molecules are in direct contact. Yes? Yes, ma'am. This is what exactly what happens and should be remembered when we talk about diffusion. For osmosis, do we understand that the molecules are in direct contact? No, not exactly. When I have to talk about osmosis, we always understand there is a separating membrane. What is the separating membrane known as? A semi-permeable membrane. Give me one example of a semi-permeable membrane. Cell membrane. Now there are so many such membranes even in our body, remember. So now when we are talking about what is osmosis then? Osmosis, I'm saying that these are water molecules, higher concentration this side and lower concentration this side. So the water molecules are crossing a particular membrane to go the other side. In case of diffusion, was there any membrane we were talking about? Nothing. And in case of osmosis, are the molecules in direct contact? We may talk about 2-3 types of molecules also here, but only the water is moving from a region of high to low. Are you understanding?
Yes.
So here, that's why we wrote what is diffusion? Movement of the molecules from a region of high concentration to a region of low concentration when the molecules are in direct contact like this. You talk about the molecules of the incense stick and the molecules of the air. They are in direct contact, no? Yes. And moving from a region of high to a region of low. Any other example this way? What is happening here? The molecules are actually crossing a membrane and then going the other side. They are crossing the membrane and going the other side. Osmosis. Right now, what's the definition of osmosis? The movement of... First of all, write down one example of diffusion on your own. I asked you example with example to write. Write down one example of diffusion. We had discussed 4-5 examples. Write one example of diffusion on your own.
Yes, ma'am.
Yeah. One example of diffusion. Write down. Done, all of you? Osmosis. Now we take the definition of osmosis. The movement of water molecules. Movement of water molecules from a region of high concentration to a region of low concentration, from a region of high concentration to a region of low concentration through a semi-permeable membrane. Through a semi-permeable membrane. Through a semi-permeable membrane. What example for osmosis you can write?
Filtering of water, like RO filters.
Filtration of the water. Filtration of the water.
They do use the process of osmosis.
Filtration of no, but for the process of, but for the filtration of the water, the ions and the molecules are also moving. No? Ions and salts also they are getting moved. So for osmosis, you always have to write examples where you are definitely writing about only the movement of water molecules.
Soaking of lentils.
Absorption of water by
Lentils and
raisins.
rice.
Absorption of water by lentils and grams. Absorption of water by root cells. All these are examples of osmosis. Absorption of water by the grams and the lentils. Absorption of water by raisins. Absorption of water by the root cells. All are examples of osmosis. Now look into your volume, all of you. Look into your volume. We'll have to solve some few questions. Let us get started with quite a number of these questions. First of all, move on to page 92. Did we do this exercise earlier?
Now the review question.
Did we do? No. Review questions. Page 92. Start with fill in the blanks and true false. Start with fill in the blanks and true false. Page 92. You have a doubt, you will clarify, you will ask. Any confusion?
Now, the cell wall is found in the cells of plants, fungi, and
Cell wall is found in the cell wall of plants and fungi and bacteria.
Bacteria is
What? Bacteria is what?
I don't believe it has.
You don't believe, you will believe later what you learn.
I've
Bacteria
learned
has
that
a
bacteria possesses qualities more like that of an animal cell.
No, who told you this?
My biology teacher.
Bacteria possesses some features like division, which is similar to developed cells. But since it's a prokaryotic cell, it has major features like those primitive cells, like possessing the cell wall, no cell organelles, etc., etc. Division pattern quite similar, like amoeba and all. आ रहा है समझ में? Clear? Whenever there's a doubt, you will ask. All these questions, again, we will try solving a few questions in the class because almost about 50-60% of the questions I will try taking up in the class. But all questions we will not be able to do. Again and again, I am telling you. So you are supposed to go through all the questions. Find out if there's any doubt, any confusion in any of the questions. So as and when you come for the lectures, you may clarify those doubts that you have in those questions. Okay? So we are going to solve in our time as much as possible, but maybe we will not be able to complete all. In the text also there are some few questions that also you'll have to go through.
Now, what is the primary function of leucoplast?
What is it? Who remembers? Leucoplast helps in storage of starch and oil granules. Starch, oil.
Storage of starch.
Starch, leucoplast. It's always a colorless plastic which is engaged in the storage function.
You can just write storage is function.
What is the statement? What is the
The
entire?
primary function of leucoplast is that.
Storage of starch you can write. One
Yeah,
function.
that's what I know.
Storage of starch. Oil droplets, storage of starch. That is the function. Fill in the blanks. All done. How many are done?
Blanks.
Complete this, then you take a break. Fill in the blanks, you complete all.
Did not get the answer to 6 and 6.
Number 5.
I didn't get a clear.
Number 5. Function of the Golgi apparatus and are the secretion, modification and transport. Secretion, modification and transport.
I have a question about it.
And
The fifth one,
fifth one only I told you right
transport.
now. Fifth one only I told you right now.
sixth one. It's
Okay, sixth one. Plastics found in
no,
most.
the question you told us was the sixth one.
Achha, fifth. The nucleus in eukaryotes is separate from the nucleus in eukaryotes is separate from the cytoplasm by the nuclear membrane.
Yes, from that.
Got all? Fill in the blanks, all of you finished? We'll continue again after the break. Finish up. If there's a doubt, you will ask after the break.
एंटरटेनमेंट चाहिए मुझे भी एंटरटेनमेंट चाहिए
Where is the answer?
नहीं वो तो हर चैप्टर में लिखा होता है
नहीं सिर्फ रिव्यू क्वेश्चंस के
No, actually, answer.
एक इंस्टेंस
ऐसे गाती है
गाती
मुझे नींद आवाज सुनती नहीं अरे कुरकुरे
आप सुनने को
मुझे भूख लग रहा है इमर व्हाट स्टेप्स
हाँ मेरे को दारू भी चाहिए कोई देगा
listen
I would love some LSD
when I'm 18 I'm getting drunk the night I'm 18 I'm getting
like I need LSD and a lot of cocaine please somebody provide इसके बाद ना तेरे को भी चाहिए रहेगा तेरे को बस पता नहीं है अभी
नींद आ रही है वापस कितना बात नहीं
और तो देखता हूँ क्या मेरी तरफ देखती रहती है अरे मेरी तरफ यहाँ सामने तो बैठा थी जा मैं घूम रही हूँ मुझे मैं लिख रही हूँ उसके बाद मैं स्क्रिबल कर रही हूँ है ना वो इससे देखेगी उससे ज्यादा साइड आई करूँगी क्रिमिनल ऑफेंसिव साइड आई नहीं किसी को एक तो मैं जितनी बार पता पलट रही हूँ ना कोई आंसर देखने चाहिए I don't think allowed मुझे अभी क्या करूँगी
क्या करेंगे तुझे
मेरे को ना मैं कल परसों नहीं आ पाऊंगी मैं जा रही हूँ बाहर
जा रही हूँ मैं भी नहीं हूँ तो मैं बाद में ले सकती हूँ
मेरे दादाजी आए और उनको सोमनाथ जाने की इच्छा है तो पिताश्री हम सबको लेके जा रहे हैं सोमनाथ
नहीं कौन टेस्ट कौन नहीं दिखा
नहीं देगा पापा आपको टेक्स्ट कर देंगे
मैं बाद में लाइक मंडे या ट्यूसडे को दे दूँगी
कौन सा टेस्ट से मैटर में आवाज करांगे
साइंस
तो
सबने दे दिया लास्ट टाइम का जो भी पेंडिंग टेस्ट दिया कितना कितना कौन सी पेंडिंग है टेस्ट
मैथ्स
फक्त एक किसने नहीं लिया मैथ्स का सबने लिया हाँ लिया किसी को डाउट नहीं थे इतने कम मार्क्स मिले हैं तो
सर ने करवाया है जितना हमको चाहिए
अच्छा हो गया सबके साथ डाउट दिया
सर ने ज्यादा किसी को कोई क्वेश्चन नहीं तो उन्होंने एक क्वेश्चन सॉल्व करके दिखाया था जो किसी को नहीं आ रहा था बोर्ड पर
अच्छा, तो समझ में आया ना?
हाँ
ये सोशल लिया अभी।
कौन कौन सोशल के तुम तीन 1 2 3
Maybe.
हाँ
मैं मोस्ट प्रोबेबली आरएम तो कर ही लूंगी घर उधर से ऑनलाइन बट ये सैटरडे और संडे नहीं आ
अच्छा ठीक है, 2 दिन का ही है ना सवाल तेरा।
मंडे को श्योर नहीं है
किसके ला रहे थे?
कौन तुम लोग लेक्चर के टाइम में तो कोने में ऐसे फंस रहे थे कभी भी खाने के